Hello, good morning, good afternoon, good evening, and welcome to our last seminar for the 11th series of the International Plant Breeding Seminar. It's a pleasure today to have Dr. Ousmane Boukar from IITA talking about cowpea breeding in Nigeria. Ousmane he's originally from Cameroon. He did his bachelor in Cameroon, and then he has a master's and a PhD from Purdue University. And all his career has been in cowpea breeding. Before joining IITA, he was breeding cowpeas in Cameroon. He joined IITA in 2003 and two and three, sorry. And he has been involved in several research projects, more than 50, both as a collaborator and as a principal investigator. He has mentored several PhD and master students, and he has hosted more than 15 visiting scientists in his program in Kano. Additionally, he has led over several training sessions, and he's the author of several peer-reviewed papers. Okay, he has released since 2008 to 2022, 37 cowpea varieties. So if you're interested in cowpea breeding, Dr. Ousmane Boukar is the man. Ousmane, thank you so much. I'm going to stop sharing here, give you the floor. Go ahead and start sharing.
Okay. Hello, can you hear me?
Yes, we can hear you very well. Start sharing if you want.
Okay. Look. Is my slide there?
Looks pretty well. You take it from here, Ousmane.
Great. Thank you very much, Carlos, and good morning, good evening, and good afternoon. So wherever you are. So my name is Ousmane Boukar, as Carlos introduced me, and I am working on cowpea at IITA, which is the International Institute of Tropical Agriculture. And I am based in Kano, northern Nigeria. And today I will try to share our experience, what we are doing here for cowpea genetic improvement. And we're really very happy to be there. And this is a good opportunity for us to interact with a larger community. So my presentation will follow this outline. I will try to introduce my institution a little bit. I'm sure you have already received at least several people from IITA, but I will also talk mostly about cowpea. Then we will see the objectives of our breeding program. Then I will quickly take you through what we have done so far in trait discovery and deployment, and of course, varietal development pipelines. So this is what we are going to follow this. So a brief overview of our institute is one of the CGIAR, that is consultative group on international agricultural research. And our mission is mostly focusing to offer leading research partnership that facilitate agricultural solution to hunger, poverty, and natural resources degradation throughout the sub-African region. And our vision is truly focusing in leading research partnership, facilitating agricultural solution to overcome hunger, poverty in the tropics, mostly in sub-Saharan Africa. So the institution have 6 crops that they have a full mandate that include banana, both banana and plantain, cassava, cowpea, maize, soybean, and yam. So then we have also some crop that we conduct some research on cow, that is cocoa, coffee, bambara granite, and other small legumes and vegetables that we have also in our portfolio. But the main crop are the six first one. And in terms of, just as all the CG centers, we are research area are covering three main research area. That is genetic innovation, mostly including biotechnology, breeding and breeding resources, the gene bank system, and for resilient agricultural system research area include farming system, agronomy, plant health, mechanization, natural resources management. And the last topic area of science that we are also involved are system transformation, that is foresight, impact assessment and scaling, gender, youth and inclusion, value chain, and nutrition and health. So if you want to know more about IHA, things like that, I have a web page there that is called so, but this is kind of a brief introduction. So we go now to our main topic, which is cowpea. It's an important grain legume grown in the tropic and subtropic, mostly in about 50 countries worldwide, and very important for more than 2 million people that they consume regularly, if not daily, cowpea. The area under cowpea, I use here the statistics from FAO, that for an average, we have 15 million hectares that are grown with cowpea. And out of these, sub-Saharan Africa has 14.7 million, representing about 98% of the global area under cowpea. The production is about 9 million tons globally, and that also Africa is leading with 8.6 million tons. You can see since 1994 here that the figure showing up to 2024, we have seen some kind of increase in yield that are going through, and that makes think that we have done something, or research have done something, although you can see that mostly the yield are derived by the area, the increase in area under production. But we have went from less than 400 kilogram per hectare, now just above 600 kilogram per hectare. That is globally. So the top 12 countries in terms of area, mostly is Niger with 5.6 million, all under cowpea. Nigeria, 4.8, and Burkina Faso, 1.3. And you can see also their corresponding production. In terms of yield, you can see the yield are really low, and this is really due to several constraints that the crop is facing. We will see that in the next slide. But cowpea is really important because of its adaptability to poor soil and drought area. It can grow even with a very little rain, just about 250 millimeter per year. You can still grow some kind of cowpea. It's also tolerant to a shade. That's why we are mostly also grown in Africa in intercropping system, where it's mixed with millet or sorghum or other crop, but mostly this in the Sahel area. It's cover well the soil and conserve the moisture, control the weedings and soil erosion. It also play an important role in terms of controlling Striga hermetica, which is a parasitic weed attacking cereals mostly. When you have cowpea, they can induce their germination, and then they cannot be able to attach to the crop if there is no crop there, and then they can reduce substantially the seed load. Cowpea also as a legume, of course, increase the soil fertility in terms of with nitrogen fixation, and we have different utilization in human and animal nutrition, as we are going to show a few of them here. And we consume mostly young leaves, also green pod and fresh vegetable. Leaves are mostly consumed in Eastern and Southern Africa. In West Africa, there is very few, but they are consuming. Also, the all are really good fodder for animal during the crop season. You can see just after harvest, you can see the worm fodders are get collected from the field and removed completely from the field and taken to the houses. You can see some of them and on top of the trees, very special and very delicate things. They don't want to live because at the drier period, there is no thing to eat for animals and they rational eat this. You can see also on top of the houses. So there are some different kind of food recipes. I think the slide, I mean, the presentation that was done by Carlos in the introductory, I've shown several of these. We consume cowpea as a boil, either with rice or with plantain. And also we use the flour to do those kind of cake, donuts, that is deep, just fried and all steam. That's here, the yellow one part is eaten with sweet potato. And also we can just, as boiled grain, mix with rice or in different form. Here is doing like a couscous, like serious things of this. And more importantly, we have seen that some countries, like here in Senegal, they are using in enriching the dough and making the bread. That is like very used in school feed at the school feeding programs that to improve their nutrition. So those are the key types of utilization. But this crop has also very important number of production constraints. The number one mostly is maybe insect can be number one because even all of our farmers here, if you ask them what is their problem, they will mention firstly insect. And insect is all throughout the crop production cycle. From the seedling stage, you can feed aphid. From flowering stage, you have flower bud and pod borers. And then from at the pod stages, you have pod sucking bugs, pod borers and others. But also in the storage, you still have weevils. So throughout the cycle, we have the farmers and producers are facing challenges with insect. We have also several diseases. We have all the classes of diseases, bacterial diseases. We have few of those, might be bacterial blight and others, but we have also fungals, several fungals, more than 30, I think, and then several also viruses, including aphid-borne viruses, mosaic viruses, yellow, and all those kind of different viral diseases. We have also facing a problem with parasitic weed that is mostly striga, and Alectra striga is more predominant in West and Central Africa, while Alectra is mostly in Eastern and Southern Africa. I don't know how that happened, but this is more what we see in the ground. And we have also several abiotic stresses, although cowpea is known as a tough crop growing in an area that no other crop can survive better, but we still face some kind of drought, heat, and low soil fertility as issue. Another general as a breeders that we need to make sure that we know the information is that we do have collection of cowpea germplasm. Mostly they are in IIT. We have 15,000 accessions and coming from 65. But we have also in US and USDA Gene Bank where they have about 7,000, and University of California, Riverside, who each have about 5,000 from those different countries. So this is a map showing the density of collection across the African continent, and as you can see. But to make it work for breeders, we have developed IITA core collection that is having 2,000. That is representing mostly the 15,000 accession. But still with 2,000 is very difficult for, maybe for breeder to handle. Then we try also to define a mini core that has 374. That is kind of highly representing the gene plasma maintained at the IITA gene bank. But also our colleagues from UCR have developed, based on the exchanges of material and things that they have in their lab, they also succeed in identify and define some 380. They share very common accession. And we have also in the gene bank some figs that were developed early to work on the drought. As origin, West Africa is the primary center of origin, mostly, but the X Africa is known for the a wild species is found there, and all the basin of the Congo DRC is also have most of those signs. Cowpea is diploid, has 11 chromosomes, and the genome size is about 613 megabase per, and is highly self-pollinated, might be 1 to 2 outcrossing is coming, % of outcrossing, and it belongs to Fabaceae as a leguminous, and genus is Vigna, and the subspecies is Ungulata, and mostly the cultivated is known as subspecies Ungulata. We have genetic resources of different thread, as you can see that picture and other picture that was shown by Carlos at the introduction. You can see that we have different colors, seed coat color, and different eyes, different shape, and different other. The same for the plant type, same for leaves and other things. So all those are existing in the gene plus, and it's good for different. In terms of genomic resources, cowpea have been for long considered as one of the orphan crop also. The genomic resources were not that developed, but we made some significant improvement currently, and we have several reports on the diversity analysis, linkage mapping, and other things that are going up. And some of the genomic tools are even being used in different method of like marker-assisted recurrence selection, marker-assisted backcross, and then for breeding, and also even we have initiated some of the genomic selection. The objective of IITA, with our long-standing experience for more than 50 years, our objective keeps changing with time. Initial focus was only for maturity. We try to have a early maturing cowpea with high harvest index. Then we switch for mostly concentrated and whole soil cropping, because as I mentioned in the thing, we have also miscropping that is happening in Africa. And then later we try to introduce or to integrate some of the resistant of diseases and nematode and insect, a little bit of insect, as we're going to discuss. Then a little bit of this was started introduced in the farmer preferred variety, so that they have something that they can use. Then we focus laterally on the intercropping that are very important. As I mentioned, most of our farmers are growing the cowpea in integration. Then also we devoted our effort, as I mentioned, on insect that are getting. We work even going to hunt for resistant gene in the gene bank, including for wild species. Then striga parasitic weeds appear in the continent, and we also the program has lived for a while on striga resistant and Alecta resistant crop. Then we realized that most of the farmers that are growing cowpea also have animals in their garden. So we tried our best to also focus on dual purpose variety that include high biomass, but also grain for feeding. The current objective is focusing mostly in accelerating development and delivering of high impact varietal product tailored to the needs of farmers and market. This is really a great change in our work system, and we try to move from the breeding system improvement to product focused delivery. We are targeting having a yield more than 25% yield over the local check that they have. And then we effort are making sure that we respond to the need of the consumers and producers. Then we are targeting adoption-driven variety system. Our focus mainly is now on West and Central Africa. So we have 4 different breeding pipelines. It is very important to organize the breeding program so that we try to respond to those needs. The first one is the short duration boiled grain and dual purpose cowpea. That is a pipeline that's targeting mostly the big market segment here with 1.5 million hectares. That is for short duration white boil grain. That is for mostly in West Africa. This is the biggest, but we also have some dual purpose for the short duration that has a significant area that we also consider in the different strategy that we are having. The second breeding pipeline is for medium duration boiled grain and dual purpose cowpea. Here we have a very big market segment that's cover 3.2 million hectares. That is medium duration white cowpea for boiled grain again, but also we have 2.7 million hectares for dual purpose cowpea. So these are the two markets in this under this breeding pipeline that we trying to develop variety that will be able to be deployed in that areas. The third breeding pipeline is medium duration homemade flowers, about 1.7 million hectares. And this is for medium duration white cowpea for homemade flour, like those donuts that I show or those steam. And then there is one that is doing mostly breeding pipeline that is addressing the short duration again for homemade flour that is having 1.2 area on the development, on the covering. So the key thread, most of them and the most to have threads are here listed. The 100 seed weight mostly is kind of trying to give us a shift between boiled grain and homemade flowers market segment. The boiled grain market segment require larger seeded cowpea. Largest for us is 100 seed weight is about, for 100 seed, the weight is about 20 gram or more. So then, of course, yield either on station, on farm, on that stress are a key. We have macrophomina in the area that is a little bit wet, but aphid in the area that are dry. We need to have those thread there so that the variety can grow. And I was talking about the dual purpose. We need also some folder. And for flower use, there are important market is key thread is easy to dehull. There are some seeds that are easy to dehull. The seed coat can remove easily, but also we have flower volumes that we are targeting variety that have more potential to have more flower when they are grand. And then, of course, maturity mostly for short duration. But beside that, there are several threads that are also a threshold that we make sure that in the background are being considered. Cooking time, for example, striga resistant, and flower thrips, just to name some. So in terms of threat discovery, so this threat discovery and deployment, this mostly what is my friend Patrick is doing here in IT. We have a global strategy that we define. Mostly we are using the stage gate system, and in stage 1 and 2 is mostly where the threat discovery activity and threat deployment is happening. Mostly we have pre-discovery, that is finding genetic variations in germ plas, identify donor parent, and then we also work on conduct some marker trait association, linkage mapping or GWAS and other study for allele discovery. Then we validate those and then make the deployment possible either through forward breeding or marker assisted backcrop activities. So those are what we have. We are kind of fortunate to have kind of mid density with about 2,600 SNPs. We have our low density is about 17 for our QA, QC genome, but we still have also several that we are kind of improving the site. So the low density is really like we're showing here is going to, in fact, being used mostly in the line development as we are involved, or the mid density also at the early staging site that we can involve marker activity. So those activities are going to be fully integrated into the breeding program. And our colleague, we show some of the activity that we have so far done. Several of them. First is to know the genetic diversity we are having and population structure, not exactly how the variation, genetic variation are in the germplasm that we're handling. Here is one of the students in our lab have done, imported some seed from Congo, about 400 of these, and study, and we can see that there are different kind of variation that are involved in that collection. Also, our mini core that I was mentioning that the UCR developed, we have also conducted some kind of analysis there, and we can see that there are several clusters that are forming, and the population structure have at least 6 clusters that have different kind of germplasm that are available for us to tap onto our Africa. Some of the other QTL analysis, I will go quickly on those. There are several of those. I select few that are more recent. We are working also in kind of identify QTL for some phenological data and for mostly phosphorus use efficiency. We have identified several QTLs either through a single environment analysis that have explained about most of them more than 10% phenotypic variation. There are some in multi-environment analysis also, 23 QTL were identified, and all for the different threads that we have incorporated in our thread. So those, we have also identified candidate genes that are underlying the QTL region that will help us to refine them and then try to deploy this in the actual breeding program so that we cannot just stay at that level of QTL. Recently also we have published this paper with our colleague from Minnesota University. This is identification of genetic variation associated with heat tolerance in cowpea. And this is mostly done with environmental GWAS analysis. We use particularly our core collection that we saw more than 2,000, and we have identified they are use some of the climate variable in the area where they were collected and conduct those environmental GWAS, and we have identified several of the variants that are associated with the adaptation to higher temperature. And we have found that the region, actually the Sahel region, which is a dry area, is a source of many of the variety that have those haplotype in their background. And those can be used as donors that for us to try to make effort in developing heat-tolerant varieties. Sraga is important. I mentioned it. We have published recently also, last year, a study where we have find very high SNPs associated with Sraga, about 5 of them at chromosome 11, 10, 2, and 2. And these were converted into CASP, and we have those markers here that are undergoing validation currently. And we hope that out of this, we will have several that we will use routinely in our breeding program. And it's really important for us here because in West Africa, at least, all varieties that's supposed to be released must be Striga resistant. So there's one kind of marker that once you work is going to be helpful in their screening. We have also worked intensively in trying to consider resistant to aphids. This is also have been done. We have identified region of the chromosome 10, 8, and 2 that have measured region that are associated with the resistance. And we have found also several genes in that region, and we are trying to involve. And actually, we have a population that is undergoing fall breeding using these CASP data. So we have also several other QTL there for seed code, which is also important for like a preference for farmers that required either white cowpea or brown cowpea, depending on the region. So this tell us that we need also to exploit that, but we have seen that there is a very complex threat, and you can see the inheritance is complex. We have seen about 116 significant digenic interaction, and also all up to 12 candidate genes were also identified for the pigmentation, and we are targeting or expecting conducting some mass-based cloning of those genes for marker-assisted selection. So also for QTL mapping for yield and other related threats, that is also ongoing. That is one of the jobs done by also some of our graduate students that have identified 7 measure QTL for 100 seed weight, number of days to flower, number of days for number of pods per plot, and other component of the yield. So putative genes were associated with yield, and then related threats have been identified, and then we are trying to validate those also probably to help us. So to summarize the threat discovery, mostly activities, we are making progress. We have 3 categories, the drought and Maruca damage. Maruca is a pod borer and post-sucking bot. We are just at the level of stage 2, which is mostly phenotyping, donor discovery. We have identified some already, and we are trying to see where to move to the next stage. We have at stage 3, most of the other threats that are very important for our different market segment, that are striga, resistant, cyst site, grain color, trip, and heat. We are at the level of locus identification, QTL mapping and validation is happening, and this is where from there we go for deployment. The one that we are on that deploying now to develop some of the pre-breeding lines and other that are going to be used in marker selection is mostly bacterial blood and aphid. So this we have already confirmed. We have validated the SNPs and others and being used for system. So those are kind of summarizing what we are doing. I left a lot of small activities that I didn't report. In terms of line development pipeline, is also we are focusing in different, that I show you the different pipelines. And for each pipeline, one of the key activities that we start when we want to implement our pipeline is, of course, parental profiling. Key parent, mostly high, allied line or things, are get profiled, including genotyping. We conduct genotyping, but also all the characteristics are there and are being recorded. This is really important for us to identify those parents and the combination to get to make combination that will result in the target threat that we are aiming to conduct. One of the things that can help us on doing that also, we are doing diversity analysis at the beginning of each set of activity we are conducting. And that also help us to identify those set of material that they have, how the genetic variation are in between among them, and then rightly said that we can select parents, and it's going to also make a strategic in terms of a number of crosses. For example, we cannot select the same parent, a lot of those parents in the same cluster, and we can say 1 or 2 representatives that can be mixed with the others, but at least, or avoid to make crosses with people within the same crosses. So those are kind of strategic. So we have, I put just a little bit of figures to help us understand the size of our program. We are not that big program, but we are handling several varieties, several sets. And the nursery, here the number of parents per year goes to up to 100, and several crosses are being done, and we generate some F1s, then those are going to F2s, and then F3 and F4. We will discuss later how we are advancing those population. So and then when they reach homozygosity, of course, they enter into the initial trials evaluation. We have an initial trial evaluation. Here is to give you how the size of the entries in that set. In 2021, for example, there are about 2,000, but in 2025, we have about 5,000 that are being tested in 2 locations, single rep, and in augmented design. So then the baseline selected from here, the advanced in preliminary yield trials. And those also, you can see the number of sets we are handling and the number of lines at that stage. And we increase a little bit the number of location, about 5, 6. And basically for prelim trials, we have 2 replication and usually raw column design. But in some cases, when we miss seed and things like that, we do a partial rep, P-reps. So that is also be done in advanced line, advanced breeding line, the base of a prelim line into the next year in that set of material that have several also. This is the size, about 300 line to 400 per year in that stage. And these are going to about 10 locations across Nigeria, but across also the continent at that point. And it's quite important that we are also in that location. I didn't mention, but we are keeping, I didn't show, we're not going to be showing my tribe, but we have a different agroecological zone that we make sure that we have those varieties are being tested so that we can know their adaptation on those areas. Those are 3 rep, and we also use most commonly row and column design. And from that, the baseline stable across location and into what we call our Cowpea international trials. Those trials are the trials that are combined, compiled, and arranged so that it's going to be help. We share with partner any request from anywhere in the world. I think we receive even from Asia, from I mean, from of course from US, but mostly in Africa. We share those with our collaborator or people that are needing. Some are using for in a breeding program. Some are also using as studying, things like that. They request those set of materials and they test them, and what is feel stable for them, they can release as a varieties. The others are just maintained as a parent in the system so that they can use. So that is common, and that's why I will show later the variety that are again released, and that is. From here, mostly in Nigeria, we also participate. Some of our line we nominate in their national performance trial. And basically, so far, I mean, we as a CG, we do not release variety. There is nothing left, but we release our variety have been released through our partners. Those are the national agricultural research services that are across the continent. Here is quite a kind of, I was telling you that I can show you how we are advancing those lines. Most of the time, and in general, we are using single seed descent that we found appropriate and easy to handle for all of materials. This is for one of our pipeline, that is the short duration. We have about 40 crosses that we conduct annually, and we have generated about 20 F1s. That is for 40 crosses, about 800 seed. We save them. We have about 10,000 seed, about 250 F2 per cross, per family. And it can be some kind of attrition, 20% we put. In general, we have more. Sometimes some cross we have less. There are also, as you can see, you have seen the number in the other slide. It's not exactly that, but this is our target. We aim to have about 250 F2 at least for each of the population we are handling. And then at that point, F3, we have about 8,000. Then F4, we conduct in an area that is a little bit kind of testing, but just washing an area where there is a striker, just observation rows that then, and then from there we harvest the best, our harvest here. And F5 is there that we start our small yield, what I call initial evaluation trials. And then the best are going to be in the prelim. Then we go to about 300 for those 10,000. About 300 is coming to advanced testing. And then we have either in CIT, that is the Cowpea International Trial, or with line that we nominate, for example, in Nigeria, there are about 30, and they are split by what you call maturity group here, particularly short duration, but so we nominate that, and there is expectation to have at least 3 or 4 out of these that undergo on-farm trial and eventually release. So those are the things that we were using for several years. And then now that we are really focusing on making sure that we improve our genetic gain, then we are thinking now, for example, to completely maybe start the initial evaluation at F4. And this is not actually a real evaluation, but it's just like that observation. Then the next from here, F4, then is going to be directly to prelim. So at least we are going to remove this cycle. And at the end also, in terms of going the advanced line, from the advanced line, we should be able to nominate lines that are going directly to national performance trials and then release. So those are really things that we think that when implemented fully, we're going to increase our genetic gain in terms of things. So the testing strategy I've already mentioned when I was talking about those figures, like initial evaluation is just mostly augmented design, about this number of lines, 3,000, and we have 6 checks, very important. Those are checks, including we have checks for maturity, and we have checks for the release variety, and also checks if in that class, particular market segment, there is some preferred farmers variety that are there. So all of these are used as a check in that line that will allow us to continue testing. And then so on, as I mentioned, prelim, advanced national yield trial, then on-farm. On-farm, you use usually RCBD. So here is an example of recently released data that I want just to show at least some kind of data, show you the feeding of the variety. The average of those 2 varieties that we released is about 1.3 and 1.2. You can see that in some locations they are about 3 tons. That is really high, but across locations, some locations are very poor. And it's still for us, we need to test them in that location to make sure that at least our farmers with minimum management skill can have. So those 2 varieties have more than 25% yield advantage compared to a check, which is here, one of the varieties that we released in 3-4 years ago, and is here. And this is the average, and you can see what we have. So that is kind of just quick example of how we are getting some dose of data that are helping us. Here is a same line tested in on-farm. Those are farmers across 11 locations in 3 states of Nigeria, having access to that. And they tested alongside their release variety, their preferred varieties, and some other potential line. But they found out that those are most highly yielding, and because of that, we succeeded to release them actually. So here is those are 4 that we have released recently. Mostly they are line from IHA released by our partners, IR. We have our standing is mostly on the yield side, but also the maturity. We have 2 that are short duration maturity and 2 that are medium maturity. So those are the line that we have in 2026 release, and we hope that we will try to push them to go to the farmers. Otherwise, I listed here, I will not go through several of release varieties, also through our NARS partner. For example, those 4 are the same that I was talking. In 2022, we have released 2 in Ghana, 2 in 2018, 2 in Nigeria, Ghana again, South Sudan, Nigeria, because we are based, several times we released there, and Tanzania, Swaziland, Sierra Leone. So some of the area are far from us, but it's still our material is being tested and released. And in fact, some of the release, we even hear it after if be approved, we read sometime we are find some of the collaborators, they inform us. Many of the time we just see either in the publication or things like that they are released, but we don't have even know about this. So just we recently, 3 years ago, we started also to see how we are doing in terms of progress. We have estimated our realized genetic gain, and for the short duration for yield, mostly here I presented all data with yield. You have a publication here that has yield and other traits. We have seen that for the short duration, we have about 2.6% genetic gain, representing about 22 or 23 kilograms per hectare per year. For the medium duration, we have few, very 0.8, representing about 8 kilograms per year per hectare. For the late duration, we have also about good level, 2.5% genetic gain. That is about 22 to 23 grams per year per farm. So that is term of realized genetic gain, but we have also used some analysis as a publication to show also some of the predicted gain that are quite high, 33 kilogram, and then for short, medium is 28, and late is 35. And here is for 100 seed weight, that is not that high, 0.8 and 0.6 and 0.5. So those are what we thought that we measure, and from here we are going to annually estimate, make sure that we are on track and follow the market, but only also make sure that the progress has been registered. So that's almost done. And I would like to take that opportunity to thank several of our donors, mostly the Bill and Melinda Gates Foundation, now on Gates Foundation. We have also benefited for a long time through USAID, Feed the Future program. Those were important for us, and they really support us on those activities. Otherwise, in terms of collaboration and several projects we are having with the CG as Excellence in Breeding, but also several universities in US, UC Riverside, UC Davis, and also University of Florida. We are having exchange of materials and different collaboration in terms of cowpea genetic improvement. Of course, we have several programs in the GC that we do. And we have also Bayern that here have been involved in kind of when we started initiating our modernization, our breeding program, we are trying to move from what we are doing in the public sector to try to emulate what is happening in the private seed company. Here are several winners that over the years we are working together. Those are the people that get at the end of taking over our variety and things like that, and this year to thank them, and we hope they will continue conversing. So thank you all for listening, and I think from here, I think we should be able to take questions if there are clarification
so
or
much,
contribution. Actually, I want a lot of contribution from you. Thank you.
Thank you so much, Ousmane. Everyone stop sharing so we can, there you are. There you are. Yes, thank you so much. A very nice, excellent presentation. I was gonna ask you about the BT cowpea. What's the status of that? It's been released in some countries. What is the prospects regarding the consumers themselves? Okay.
Yes, thank you. So the BT project, IIT was involved in the beginning. We have designed a lot of those events. We have struggled to succeed. Later, the partners in Australia have able to make transformation because the bottleneck actually is transformation. And then from there, there was a lot of other interaction partners. And then since we are not going to release them, most of the activity were conducted with our partners here in Nigeria and in Ghana. So in Nigeria, in collaboration through those partners, they have succeeded to release one, and a second is coming. One BT cowpea, and is getting commercialized with farmers. We had experience because the farmers have received might be not good information. they were received that this variety should not spray, no insecticide, while the BT is just controlling the pot borer, is not controlling the other insects. So the first year was a little bit not good, but later that they understood what is the target, then I think they are getting in and things are getting on. And colleagues are really now trying to go with a foundation, get foundation part of the Act One, they call them alliance. They create alliance that, and they are pushing in high level support that they are supporting them. And Ghana is coming for release soon. And I think there is perspective in West Africa at least for this product.
Yeah, it's a very interesting case of biotechnology trade that is being commercialized in Africa. Okay, so
yeah,
it's very good. I have a couple of questions here in the chat. Why do you think is the lower genetic gain in the medium maturity?
Why is the lower genetic gain?
Yeah, the lower estimated genetic gain that is 0.85 compared to.
Oh, for that medium. Okay. Yeah. Yes. So in fact, the analysis were done for 10 years. We selected the last 10 years and we conducted, and we selected common area. So it's happened that in some of those common area, quality of data is one, but I'm not saying that, but maybe also our varieties in that segment are not performing higher than the checks that are there. This can be one of explanation. Other explanation that I see, I think mostly is going to be also in terms of really genetic gain, in terms of threat that are related to yield. That is number of feed. When the maturity might be is not is at the medium size, I don't know, maybe there is a kind of affecting those production threat or something like that. But anyway, it's mostly in terms of the difference between our advanced line and the checks that we are having at that price. Maybe we didn't make a significant difference.
Okay. What is the potential for gene editing for some of these traits, and is it being investigated?
Absolutely. We, in the current project we are having, we call it a breakthrough project, and it's funded by GIT Foundation. The aim is really to explore all the available technology so that we can get that yield. I mean, we have to boost that yield in some way. We are still struggling getting to, while our colleagues in Brazil, actually, they get about 3 ton, 2 ton is really common in Brazil. But we here, we have 1.5, our medium, but if you don't treat them, then you have more problem. So I think mostly for gene editing, we started working on that. We actually, Lina, we have a lab in Kenya, Nairobi. They have started, we collected some line from us with different maturity group and different seed color, things like that to try. So we are targeting aphids, seed size. We think that those we can, that is like a quick win. We want to show something that is working so that from there we can tackle all of the other threats that are more complex. But we think that given that we have good markers and other things on aphid and then seed size, then we are working on, actually those are our two preferred thread that we want to start. Then from there, we explore something like a pot soaking box that we don't have anything about that.
Okay. Question from Abe Garano from South Africa, I think. Is it possible to release your parental lines after studying their adaptability without any crosses, or do they need to go through a crossing cycle?
Yes. So for our colleagues in the NART program, mostly they take our varieties, mostly either in the set of what we call Cowpea International Trial or the advanced line that we share with them. Then they test. When they see that it's really those materials are stable in their location and also beating their local check, their commercial check that are already being released, the last variety that is released, that they have things like that. If they release, then they already, some are asking us to give them our pedigree information, things like that. Then they make the package and release. So some of the variety are going to be very good in some traits that they also can observe. Some like maybe, I don't know, striga resistant that they observe in their area, very good. Then, but they don't like either one or two then. And that place, then they can do some crosses with their alert line to just for integration or something like that. And then they release also. So we have those examples also to cite.
What's the reason that there's such a low number of varieties released in Southern Africa? Is it because of lack of adaptation not being your primary target?
Yes, might be, but also the level of collaboration at the beginning was high. Then later we don't have too much collaboration with them, although we still maintain some collaboration. In even South Africa, some of we lost, I think actually even two of our very dedicated breeders that are working there testing some of our varieties they have released. Unfortunately, we lost them in 2 years ago or three years, maybe more, 4 years ago. Then we lost kind of contact, but we still have some people that we are interacting with them. But most of them currently, they are just looking for studying stress, studying things like that, but not really involved into release of varieties.
You know, Ousmane, I think we have talked about this. I'm very interested in Vignas, particularly for product development, new products, okay? And I told you that I have made a tempeh with cowpeas, and it's the best tempeh I have made. So there's a lot of potential there, aside from the traditional uses. And I hope one day we can realize that potential. Okay.
Yeah, that is things that we are really thinking now in terms of since we are going towards demand-led breeding. We focus mostly also in offering. So one of the big questions that we receive actually from our donors is that, okay, you people are aiming to increase the yield, and what is going to happen if you have more production? What is going to happen? So I think this question is coming, and then we are making sure that with the interaction with the processors and other things, we are trying to find new utilization to increase that perspective, so that in case we succeed really to have that enough people, which is really difficult to do, but we think we are going to get there, and we hope that we should be able to provide to the people.
All right, I took the liberty to put your email address there in the chat, Ousmane.
Yeah.
And I asked, I know there's a question or two that we didn't get to address. Please write directly to Dr. Boukar. I think he's more than happy to clarify or answer questions. The same if you need to request germplasm or see the possibility of introducing germplasm. Thank you so much, Ousmane. It's been a pleasure to
Absolutely.
have you close this series. This is the last seminar for this series. We intend to start a new series on October 15th. Please volunteer. I'll be tapping on some shoulders for sure. I think the whole set of speakers, we focus this series on African colleagues, African plant breeders. It's been amazing. I mean, I learned a lot and I have enjoyed it a lot. Okay. The link to the recordings, we're going to send the last seminar, and for all the seminars, the link to the recordings is going to be at the bottom of that email. So please take a look at them. And later on, after we do the editing of the captioning, they will be posted in YouTube for anybody to access the seminars. Okay. Thank you again from here from North Carolina State University. Thanks, Brando, for your help as our IT support. And we look forward to seeing you again towards the end of the year in October, okay? Thank you so much. Have a great season, okay? Have a great season in the field, Ousmane.
Thank you. Thank you very much. I appreciate it. Thanks.
Okay. Bye, my friend. Bye.
Bye.